Okay, welcome to Chapter 4, where we discuss sex determination, that is how sex is determined in offspring, and sex linkage, that is genes on the X and Z chromosomes. First, we'll discuss sex determination, and you'd be surprised at how many ways there are determining sex in animals. Then genes on the X chromosomes, then genes on both the X and Y chromosomes, patterns of inheritance of sex-linked genes, and lastly, dosage compensation. So the basic animal life cycle, where fertilization restores the diploid state and the zygote grows via mitosis into an adult that produces gametes via meiosis. And of course, those gametes are haploid, as we know. This shows the size difference between the two gametes, a sperm touching the surface of an egg in the early process of fertilization. So there are genetic and environmental ways that sex is determined. One genetic way is haplodiploidy. There's XXXY, ZZZW, XXX, and a compound system where the sex chromosomes are in groups. Then there are environmental ways like temperature-dependent sex determination, sex of nearby individuals, and infectious agents can change the sex of some organisms. So we'll discuss the environmental ones first, and then the genetic ones. So in many species of reptiles, including all crocodilians, most turtles, and a few lizards, is temperature-dependent sex determination. So in turtles, the temperature that the eggs are exposed to during the middle trimester of development determines the sex of the little turtle embryos in those eggs. And in one species, temps of 26 to 28 degrees C produce males. Higher temps from 29 to 34 produce females. There are three different ways that can be done, where MT refers to male temperature, FT female temperature, P parity, and then the y-axis is percent female. So in case one, you get females at lower temperatures, males at higher temperatures. Case two is the reverse of that. And then case three, we get males at intermediate temperatures and females at high and low temperatures. The sex of the nearby individuals is found in a little mollusk called a slipper limpet. They live in stacks, and they can develop into either males or females. But if one attaches to a female, it will develop into a male. Isolation of a large number of males causes one to become a female, and after it becomes a female, it does not change back into a male. This shows you four of them stacked together. The bottom one is undoubtedly a female, the other three are probably males. And this shows you kind of how it works. When a larva settles onto an unoccupied substrate, it develops into a female, and then it releases chemicals that attract other larvae, and they settle on the female and become males. But if the female on the bottom dies, then the ones above her develop or turn into males, after which they don't change sex again. Infectious agents, isopods, or little terrestrial crustaceans. In Europe, if they normally have a ZZ-ZW sex chromosome system, but if they are infected with some bacteria in the genus Wolbachia, that changes the males into females. Here's a little guy here. We call them in this country roly-polies or pillbugs or sowbugs. On to the genetic sex determination mechanisms. Haplodiploidy is found in social insects like ants, bees, and wasps, where the females are diploid and the males are haploid. And this shows female worker bees. The workers are sterile females, usually daughters or nieces or sisters of the queen. So the queen will mate, and she can store sperm, quite a lot of sperm, and she lays eggs, but when she runs out of the sperm, of course, what will she need? And that's males. And so she starts laying unfertilized eggs, which develop into males. And males make sperm via mitosis and not meiosis. The XXXY system is found in us mammals, Drosophila fruit flies, and Central American musk turtles. The females are the homogametic sex. All their eggs have the X chromosome. Those are the heterogametic sex, so roughly half our sperm carries an X, the other half a Y. So it's the heterogametic sex that determines the sex of the offspring. And our Y chromosome is only by maybe a third or so the size of the X chromosome in us mammals. And so this shows you sex determination in humans, where a male produces roughly half his sperm carrying the Y, the other half carrying the X, and that determines the sex of the offspring. This shows you again, the X is about 2.5 times the size of the Y chromosome. A human X and Y next to one another. They do synapse together. This is an X and Y chromosome in the mouse, and you can see that the Y is just this little segment here that sticks up, and the X is the big long bent chromosome. This shows you that in the presence of the SRY gene, which is the sex-determining region of the Y gene, it produces a protein called testis-determining factor, or TDF, and that causes the medulla, or the inner part of the embryonic undifferentiated gonad, to develop, and that develops into a testis, producing testosterone, creating the male. In the absence of the TDF, the outer layer, the cortex of the embryonic gonad, develops, and that develops into an ovary. In the absence of testosterone, develops into a female. The ZZW system is kind of the reverse of the XXXY found in birds and many lizards and snakes and those isopod crustaceans. Here the female is the heterogametic sex having Z or W chromosomes. The male is ZZ, so therefore the female determines the sex of the offspring. Roughly half her eggs will carry a W or half a Z chromosome. This shows you how that works, where the male here is ZZ and the female is ZW. The XXXO system, females have 2 X chromosomes or 2 sex chromosomes, where males only have 1. And it's found in some insects. So males produce gametes with a sex chromosome or without a sex chromosome. So therefore, the males always have 1 less chromosome than the females. So if the female is 2n equals 20, the male is going to be 2n equals 19 because he only has a single sex chromosome. And this shows you in the upper panel how that works, where the male has 1 X only, the female has a pair of X. So the male determines the sex of the offspring by whether his sperm carries an X chromosome or carries no sex chromosomes. In this compound system, one example is from a nematode worm called Ascaris incurva, where the male is 2n equals 35, 26A for autosomes, 8 X chromosomes, and a Y. The female of the same species, though, is 2n equals 42, with the same 26 autosomes, but 16 X chromosomes. So in this species, X chromosomes occur in sets of 8, not 1, but 8. And they unite together during meiosis and behave as 1 unit or 1 large chromosome, 1 pair of large chromosomes. Now in the duck-billed platypus, a mammal, their sex chromosomes occur in sets of 5 and not 8. So females have 10 X's, males have 5 X's and 5 Y's. And this shows you how synapsis occurs in a duck-billed platypus, where the different X's and Y's can synapse with each other based on these similar colored little bands that indicate regions of homology among the chromosomes. So this is table 4.1 for your text, just reviewing some of these things, the sex determining mechanisms. Interestingly, sometimes they can interact. The introductory little story at the beginning of chapter 4 talks about the bearded dragons from Australia, which normally have a ZZ-ZW system, but sometimes that can be overridden by temperature-dependent sex determination, where we get ZZ females that actually can produce more eggs than the ZW females. Quite interesting, I think. Okay, moving on to sex linkage then. These are genes located on the sex chromosomes. There's more than 1,000 genes occur on the human X chromosome. Holandric genes are genes on the Y chromosome, and the Y is 164 million bases, where the X is the X is 164 million bases, where the Y is only 59 billion base pairs, with only about 150 genes on it. Some important terms are heterochromatin. This is inert DNA that does not decondense into chromatin during interphase, doesn't have genes, tends to be around the centromeres of the chromosomes, where euchromatin is the rest of the regular DNA that has genes and can decondenses into chromatin during interphase, etc. So here's an X and a Y. Again, the X is about 2.5 times the size of the Y. Here are a few genes mapped to the X chromosome of Drosophila, the fruit flies. Landric genes then, genes on the Y chromosome. So the Y in humans anyway is about 2/3 heterochromatin, so most of it has no genes at all. But it of course contains the SRY gene for determining the sex of the offspring. And if you take that testis determining factor protein and inject it into XX mice, they become they become males. This shows that the sex determining region in the Y chromosome is on the short arm, this little blue band here. Now, pseudoautosomal genes are genes that are found on both the X and the Y chromosomes. These are little regions of the sex chromosomes that are homologous to each other. So remember the X and the Y synapse during meiosis, but of course they can't undergo crossing over because that would yield some messed up chromosomes. And some examples of genes that are the pseudoautosomal regions of the X and Y are zinc finger gene at ZFY, MYC2 antibody gene, and that sex determining region of the Y chromosome. So here we see our primary pseudoautosomal regions are the ends of the short arms of the chromosomes, and the secondary ones are the ends of the long arms of the chromosomes. So now let's look at patterns of inheritance of X-linked genes. So remember, we males only have a single X, and the alleles on that go to our daughters. Our sons get the Y chromosomes, so they don't get any X-linked traits from their fathers. But we males get our X-linked traits from our mothers, because that's where our only X comes from. We call this a crisscross pattern of inheritance, because traits from one sex move to the other sex in the next generation and then back again in the following generation. Sex-linked genes also show non-reciprocity in crosses, and I'll tell you what that means momentarily. So this shows at the bottom panel then how the sex chromosomes are inherited. The little green Y chromosome goes from father to son, and the X chromosomes go from father to daughter and mother to son. So here are two reciprocal crosses for a X-linked recessive trait for white eyes in fruit flies. Red eyes are the wild type, and now our alleles are superscripts on the X chromosome. X plus is wild type, plus always means wild type, and plus is always dominant. Wild type is always dominant. XW is for white eyes, and that's recessive. So in this cross, we start on the left, we start with a red-eyed homozygous female and a wide-eyed male. And the other one, we have a wide-eyed homozygous female and a red-eyed or wild-type male. So we can see we're starting with different numbers of copies of the XW chromosome in these two reciprocal crosses, which is going to yield different results. So in the F1s, we see that the cross on the left, all the flies are wild-type. The cross on the right, only the females are wild-type, the males are wide-eyed. And then in the F2, we see that the males show a 1-to-1 ratio of wild type to wide-eye, and all the females are red-eyed. But in the cross on the right, both sexes show that 1-to-1 relationship of wide-eyed to red-eyed flies. So we can see different results in these reciprocal crosses. We don't see this with autosomal genes. And here, both those crosses side by side. And let's look at a similar thing with birds. So you're looking at peafowl, where ZCA plus is for blue, and it is dominant, and ZCA is for cameo, or I guess a brown, which is recessive, although the blue doesn't show up in the peahens, only in the peacocks. So here we take a blue female and cross her with a cameo male, and then reciprocal, a cameo female and cross her with a blue male. And we see differences already in the F1s where the males are blue in each, but the females are blue on the right, cross on the right, and cameo on the cross on the left. And then in the F2 generations, we see that the males exhibit a 1-to-1 phenotypic ratio, as do the females in the cross on the left. But in the cross on the right, the females exhibit a 1-to-1 ratio, but the males are all blue. And here are the two crosses side by side. Lastly, dosage compensation deals with how do organisms compensate for this difference in dosage of X-linked genes, where females have twice as many as males. Well, in mammals, one X chromosome is inactivated in every cell during embryonic development. This inactivated X chromosome is referred to as a Barr body, named for the guy who discovered it. And it's a dark staining body in the cell nucleus. And it's really a tightly coiled heterochromatic X chromosome that's been inactivated. We see the arrows pointing to Barr bodies and normal human female cell nuclei. Normal human male cell nuclei lack them. This table shows that no matter how many X chromosomes you have, only one X will be active, the other will be inactivated or made into Barr bodies. So the top row is a normal female, followed by a normal male, then somebody with Turner syndrome, meaning monosomy for the X, only having one X, and then males with Klinefelters having extra X's. They're still males because they have the Y though, and then females with extra X's. And I'm sure most of these individuals with several X chromosomes are very, very rare. You're probably not going to meet someone like that walking down the street. So there's a region on the X chromosome called the ZIC, and it contains several genes that are involved in X inactivation. And ZIST is the gene that starts the inactivation process for the X chromosome. But it turns out closer scrutiny has shown that there really are some active genes on the Barr body, and some of these are even in the pseudoautosomal region. In fruit flies, which also have a XY sex-determining system, dosage compensation is achieved differently. Here, the male's X becomes hyperactive, producing as much RNA as both the female's X's. So this shows that X inactivation occurs during embryonic development in mammals. In humans, it occurs 12 days after conception, but the same X is not inactivated in every cell. So if a female is heterozygous for an X-linked trait, she will show different traits in different cells, what we call mosaicism, the cellular level. Calico cats are a great example of this, as are tortoiseshell cats, all being females. Women can have something called ectodermal dysplasia. If they're heterozygous for that, that means they have patches of skin on their body with no sweat glands. So this shows you in a calico or tortoiseshell cat, you have one X chromosome that has a gene for brown or black coloration and the other for yellow or tan coloration. And depending on which one is inactivated, those cells will exhibit different colors from each other. And that's how you get the different patches of fur color. So here's a calico cat showing you to the left there, the nucleus with the Barr body, and then the cat on the right. The white comes from a white masking gene where neither trait is shown. Here's a tortoiseshell cat, pretty much same as a calico cat, just lacking the white. So this little black patch here means that it came from a cell where the X carrying the yellow was inactivated, and then that cell gave rise to a patch of fur that is black. That's how that works. Here's a female human mosaic for ectodermal dysplasia. These pink patches on her body lack sweat glands. And at the molecular level, we have an active and inactive X chromosome. The inactive one, the ZIST gene, produces an LNC RNA. LNC stands for long non-coding RNA, which coats that chromosome and allows some proteins then to bind to that chromosome and cause it to be inactive. So the one on the right with the little purple balls on it, that will become the Barr body. Here's a close-up of the X inactivation center, or the ZIC, showing you the ZIST gene there in pink, and then 3 other genes that are important in X inactivation as well. This lists those genes and what they do. I'm not going to read it for you. And then in fruit flies or Drosophila, these little balls on that male's X there are proteins that cause that chromosome to be activated and produce, hyperactivated, I guess I should say, and produce more RNA, or as much RNA as both the females' X chromosomes shown over on the right. So from this chapter, do problems 25, 30, and 35. Thanks and goodbye.